Biomedical Sciences PhD. So Nicole?
Well, hi, I'm Nicole. I work in Dr. Von Kamp's lab in the Biology and Biomedical Sciences department. I'm in my 4th year of my PhD, and I study polyamines. Polyamines are something that very few people have actually heard of, even if you are in the sciences. They're this small, positively charged group of molecules, and they're in everything. All living organisms, from single-cell bacteria like E. coli to humans, us, every living cell has these molecules in them. And they are involved in all sorts of cellular processes. One big one would be something like cell division. So when the cells need to multiply and divide, they have to have these molecules. And in order for these molecules to be effective, cells have to maintain a precise intracellular concentrations. They have the right amount of these molecules. And to do that, they have 2 methods. They synthesize them, they make them, or they also can import them, bring them in from the outside the cell. And while we have a good understanding of how they go about making them, that synthesis pathway, there's this big black box of lack of understanding of how in higher organisms like humans or multicellular animals like fruit flies, what I work in, on how those animals are able to import polyamines. So this is a big gap in our knowledge of just basic science, something that's in everything and we don't even know how it gets in. And so what I've been working on is trying to identify genes and proteins that are involved in that polyamine transport system. And I've developed a system, a testing system that lets me look in live, in vivo, meaning in a whole live animal, and see what is required for transporting these molecules, these polyamines. And I've actually successfully identified a gene that's required for polyamine transport in Drosophila, in fruit flies, sorry. And what's interesting about that is we've actually been able to look at its ortholog, its human version, and there's actually versions of this same gene in humans, and we're trying to use our collaboration with a chemistry lab and prove that it has the same function in humans, its ability to transport polyamines. And we also can use this testing system to identify other components of the transport system, and we can take that further and look at where they're expressed and other aspects of that. There's also some interesting clinical relevance, because if you think of their requirement for cell division, if you think about cancer cells are dividing rapidly, so if you can limit the polyamine pool, either by limiting the synthesis pathway or by limiting the import, you might be able to target that cancer. And if we can understand the transporter better, we could potentially use that as a target for cancer treatment. I'd be happy to talk to anybody further about that or other aspects of this at my poster later.
Thank you.